like to thank Peter very much for inviting me to this very interesting specialist workshop on Down syndrome and its potential therapeutics. I have to admit that I have worked on Down syndrome, but it was quite way in the past. But very recently, I've started working with Ben Tico on looking at methylation patterns in brain from fetal Downs. So I'd like to really tell you about, to start with, a baby biobank that we have set up at Institute of Child Health in London, because we're very keen to look at problems in pregnancy and to actually find the best way of studying them on an evidence-based molecular approach. This is just a little bit off the subject of Downs, but I think we'll come back to it and you can see why I'm going to tell you about this. The baby's normal growth throughout pregnancy is very large at the end of development, and you probably also all know that brain development is most in the last 20 weeks of development, and also after birth brain development is very important. And these kind of growth profiles are very important when you're looking at studying growth in utero, and in fact any development in utero. And we are very interested in looking at the trimesters of pregnancy and looking at the key problems in pregnancy which fall in terms of implantation, and then later on we have preeclampsia and growth restriction and prematurity. And this is a continuum, and quite often individuals that have an implantation problem in another pregnancy will have preeclampsia or prematurity. And these are the very big problems of pregnancy. And for example, recurrent miscarriage, which is one of the 4 big problems of pregnancy, is defined as a loss of 3 or more consecutive pregnancies, but it affects a lot of couples trying to conceive. In fact, 100,000 families are newly affected each year in the UK. And I just put up here a pedigree. So this is the genetic analysis of these complications of pregnancy. A pedigree of the lady in the centre with a blue circle, had in total 28 recurrent miscarriages with, in fact, 3 different sets of partners. And the key thing about recurrent miscarriage and thinking about Down syndrome is that we are very bad. Human beings are very poor at conception. We are probably much poorer than anyone can imagine, almost probably as poor as the elephant, and their gestational age is nearly 2 years. 1 in 4 pregnancy it doesn't go to term, and of those 1 in 4 pregnancies, for any individual, 60% of those are trisomies, and a large proportion of those can be Down syndrome. So actually what you're looking at is a species that's poor at conceiving, and that trisomy is very high. Trisomy in the mouse is not obscene. Mice do not get trisomy naturally. It's not a phenomenon of the mouse. I mean, we create mouse models which are clearly important for looking at human diseases, but it doesn't happen in the mouse naturally. It happens in the human. It's very common in the human. And I think to put that into the context of Down syndrome is important. Prematurity is also very common, and problems with prematurity, you can end up with poor brain development, and you end up with cerebral palsy. And 1 in 13 babies are born before 37 weeks. We're also not very good at taking babies to term. Growth restriction, in utero, the baby is born small, and this is the case also in Down syndrome. When you have 3 chromosome 21s in every cell, your cells divide poorly, and the Down syndrome infant is small as a consequence of that poor division. And we can learn a lot from growth restriction in utero, which could be applicable to Down syndrome. So, and just to end on preeclampsia, which is a problem really in the mother, and the mother can die from having preeclampsia in pregnancy. And 1 to 2% of pregnant women are affected by preeclampsia, and this is a very sad picture of a child looking at his mother's grave. What are we doing about this in London? Well, in London we have money from Wellbeing of Women, which is the charity at the Royal College of Obstetrics and Gynecology. These are all the folk that we've employed on the baby biobank study to collect samples from pregnancies. So normal pregnancies and pregnancies that are abnormal, from recurrent miscarriage right the way through to preeclampsia. We are collecting white European trios at the moment. We're hoping to extend it to other ethnic groups. We're collecting 500 of each of these trios. We're taking blood from the parents. We think the father is just as important as the mother in terms of the genetics and the effects on complications of pregnancy. We're taking cord blood, placenta, membranes. We're also taking buccal cells from the babies. Of course, as a consequence of this study, we are also picking up babies that are born with Down syndrome. We are targeting white European women. We talk to them at the antenatal clinic. We ask if they want to be part of the study. A lot of these women will not have any problems in their pregnancy at this stage. A lot of these pregnancy problems happen later. And we ask them at this stage if they want to be part of the study. We explain to them the study. It's a linked, anonymous study. We have to have it linked because we have to track the mum from antenatal through to birth. Otherwise, we will not be able to pick her up at birth. And I can see the clinical people in the audience are nodding. You know about these sorts of studies. This is what we have to do. We identify them, we give them information about the study, we explain to them what we're trying to do from a point of view of looking at the genetics and potentially the epigenetics and the proteomics and the metabolomics of pregnancy. And this is because we're taking all these different tissues and storing them hopefully in a manner that can be used by many studies, we're hopefully covering nearly all the bases that we know about technologically today in terms of methodology, in terms of future research. This is a bank. So I stress to you in Malta, this is a bank that you can have samples from for research projects that you do. For the people from all over the world here today, you can also apply and use samples from this bank. We take blood, and these are taken by phlebotomists. Our biobank staff are trained to take blood from individuals. We take urine from mum. We make serum and plasma, and we snap free samples, and we also put samples in RNAs later and in pack tubes for RNA. This is how we take from the placenta. The placenta belongs to the baby. It has the baby's DNA. It's something that the baby has made from their own genome from both parents. It's a very useful piece of tissue. It's very important for a normal pregnancy. So we take pieces of placenta to study all sorts of aspects of the pregnancy. We are collecting all these different 18 fields of medical interest by hand, and that's the staff are collecting this at the point of collection, but we're also downloading data, medical records. So if you wanted a sample set from this, say you wanted 100 controls for control studies for working with a Downs study, for example, you would get these fields, sample sets from all of, hopefully every tissue you could possibly use for your work, and you would get data downloads. So you could look and track at what happened to those individuals in the family. These are therefore anonymous at this stage. They can't be linked back to the patient, so there's no problem of misuse of data. This is just to show you the recruitment. We started in 2010. We actually started in 2008, late 2008, and we took a year to get the ethics sorted out for this. I suspect it's even more difficult in other countries, but you have to work with all the hospitals involved, all the different research and development committees, all of the different trusts to set it up. But we've managed it. It's now going. I hope you can see it's exponential in collection. These are the women recruited against the recruitment projection. And these are the women, fathers and babies. You see that the fathers are late. They are the blue bars. The reason they're late is they usually don't give us samples until the baby's born. When the baby's born, they're very happy to give a blood sample because they have seen how much trouble their wife has been in giving birth. At the antenatal clinic, they're not so keen. And then, of course, we get the samples from the baby when they're born. We hope that we're going to be able to look across these big complications of pregnancy, prematurity, preeclampsia, growth restriction, and recurrent miscarriage by looking at networks. You're all doing that. All of the scientists that I've talked to this morning are looking at networks. Very important networks in metabolism and in proteins. We used Ingenuity just to see if we could pick up some links of genes. TNF, it's in the red, it's probably not very clear, is a gene that's been implicated in the literature across all the problems in pregnancy. And these are the ones we're looking at. There's a website. I have some postcards in my bag I can give you with the website. You can go online, you can see how many samples we've collected. Very soon, in August, we're going to have We're going to open the bank to researchers so you can apply online with an application to use the samples. There's a very small cost for each sample. It's totally within budgets, certainly within budgets of anybody in the Western world. These are the sorts of publications that we have published using trio collections. So looking at cyclooxygenase, looking at all sorts of other markers of pregnancy, including genetic markers that are imprinted at FILDA2, which is work that Ben's been working on as well. Now, as a consequence of getting pregnancies that have failed, we have also collected tissues from failed pregnancies of babies with Down syndrome. And this is just a table of the tissues that were collected and the ethnicity. I think there are 10, in fact, Down syndrome failed pregnancies. You can see the tissues that were collected. Ben and I have collaborated on brain, but you can see there are other tissues that may be relevant to studies on Down syndrome, including gut and lung and so on. So you can, there's certainly the researchers in this room and other researchers on Downs could apply to use these tissues. We have maternal and paternal DNA, and we have tissues from the baby. In the past, I've published using these tissues. We've looked at different. I needed to mention, I also have collected tissues from trisomy 13 and 18, Patels and Edwards syndrome. So one could do some comparators between different trisomies in terms of looking at these very important fetal tissues. So these are babies that haven't been born alive with Down syndrome. Just one piece of work. I haven't got my glasses here, so I'm going to blag it because I can't see what's on this. I can see better up there. We looked at the trisomy signals in different tissues with Down syndrome babies to see whether, in fact, the babies that are the fetuses that don't survive had a different level of trisomy in their cells. And we found this not to be the case. It seems that they are up in the 90s for all of the trisomy across all the tissues. So I thought maybe the ones that didn't survive had a higher level of trisomy and potentially they were more mosaics that did survive, but it seems that this is something that's fairly consistently taken through development. So these are the sorts of studies that you can do. This is a slide that Ben put together. We sent Ben, Tico, some fetal brain samples from Downs and from controls, and as he described earlier, you see similarities in the genes that are methylated or not methylated going across from the Downs compared with the controls from 18-week gestation to adults in the brain. So this is saying that these changes potentially, and some new changes that go with becoming an adult, are there in the fetus and then are also picked up in the adults. So I think this is very exciting. I think a lot of things can be picked up and interrogated using this technology. I think all the people here are working to the same end, and that is to ameliorate the symptoms of Down syndrome for the individuals that we have in our societies, to make their cognitive development, their risks for different disease phenotypes lower. And in fact, there's one other thing I wanted to add to that by understanding the disease phenotypes in the Downs population, we can also understand our disease phenotypes. We are not peculiar in terms of getting Alzheimer's and getting leukemias, just because we don't have an extra 21. So in some ways, they're a naturally occurring model that if we do intensify our research on to help their situations, we can learn about diseases that affect all of us. And I think I just wanted to make that point because we're always sort of separating them out, but in fact they're a very good model for us to understand our own disease phenotypes, and so they can help us understand our Alzheimer's, and we can hopefully help them. These are just the team for the Baby Biobank, fantastic hard-working clinicians and the Baby Biobank staff. They're listed here. I also want to thank Phil Stanier, who works with me on these projects, which has been a lifelong ambition of work. As I say, we need to thank the Wellbeing of Women, who are a charity associated with the Royal College of Obstetrics, who funded this project, and Mother Care Group Foundation. Thanks.